Once the sperm penetrates the egg, it induces the secondary oocyte to complete its meiotic division. So the oocyte undergoes meiosis II and becomes a mature ovum. So now we have a haploid ovum with its set of chromosomes and a haploid sperm with its set of chromosomes. Together, these two haploid sets can come together and make a diploid zygote. We call these the pronuclei, the male and female pronuclei that merge together and form the zygote, which is the first diploid cell of the embryo. This zygote now divides. It divides into two cells, and then the two cells divide again and make four cells, and so on, until a cluster of cells are formed called the morula. These stages of division, which are mitotic divisions, is called cleavage. The final structure that is formed is called the blastocyst, and this blastocyst leaves the fallopian tube and implants into the endometrium of the uterus. So here is implantation. Here's a closer look at the blastocyst. We have the blue-colored cells forming the trophoblast and the pink cells forming the embryoblast. The trophoblast becomes the placenta and the embryoblast becomes the embryo. Once the implantation occurs, the cells of the trophoblast release a hormone called the human chorionic gonadotropin, or HCG for short. HCG forms the basis of the pregnancy test. HCG, its function is to act on the corpus luteum and maintain it. The corpus luteum, thus maintained, continues to produce progesterone. Progesterone maintains the endometrial layer. So let's go over these steps one more time. So once the embryo implants, it releases HCG. HCG then activates the corpus luteum and maintains it. Corpus luteum releases progesterone. Progesterone maintains the endometrium, and once the endometrium is maintained, there is no menstrual period, and the woman is officially pregnant.